Yeah. Okay. So Dr. Giri is a recipient of Swan Jayanti Fellowship, which is awarded by DST Government of India. And Dr. Giri has an honor of being the first plant scientist to be selected for this fellowship. So it is a prestigious award. So Dr. Giri with his team is currently focusing on finding novel genes and understanding their functions in cellular signaling in response to limiting phosphorus conditions. So Dr. Giri has adopted the state-of-art methodologies and genomic technologies such as transcriptomic, metabolomics, phenomics, transgenesis, and gene editing techniques to identify different candidate genes for improving plant performance in low phosphorus input systems. Dr. Giri has been previously awarded with Professor Y.S. Moti Medal for Young Scientist in 2019 by Indian Botanical Society and Professor Hiralal Chakrabarti Award in 2019 by Indian Science Congress. He is also a Nasi Scopus Young Scientist Awardee in 2018, and Indian National Science Academy Medal was awarded to Dr. Giri for Young Scientist in 2015. He is also Young Scientist Platinum Jubilee Awardee from the National Academy of Sciences, India, and DBT Crest Fellowship Award for 2012 and 11 and 12 from DBT India. So a big list of awards and recognitions, they are associated with Dr. Giri. So many of the other celebrated fellowships are also granted to Dr. Giri. That includes JRF and SRF fellowship from CSIR. He's a university gold medalist for first position in master's examination. So Dr. Giri has successfully published his work in many reputed peer-reviewed journals and SCI index journals such as Nature Communications, so which is a big platform to represent the research that Dr. Giri has carried out. So also in protoplasma, plant physiology, plant scientific reports, etc. Dr. Giri has to his credit several scientific projects also which are funded by different government or agencies and scientific departments. So recently also Dr. Giri has been interviewed on popular science program Eureka, which is conducted by Vigyan Prasad. So with this and many more, which I might have missed, Dr. Giri, so I welcome you on this platform to share your experiences, your research about CRISPR-Cas and genome editing with our faculty members.
Thank you, Dr. Meghna, for really kind words.
My pleasure.
Yeah, and thanks for inviting me here. There is a slight change in the program. Although it's not a change, it's an adjustment. And I want to be honest with you. Actually, I had a emergency meeting scheduled at 2:30. And I have to attend that meeting, and that meeting might take more than an hour or so. So to make sure that I do not miss my lecture, I recorded my lecture this morning. In fact, hurriedly.
Oh, wow.
So, because I have to do both the things. So lecture I will present to you, but because the meeting is online, so I am also sitting here. So if there is any issue, I am sitting here.
Definitely.
I'm not sure at what stage in that meeting I would be towards the end of my lecture. I'll try to answer the questions. But as a backup plan, I have requested two of my postdocs who are actually doing the gene editing work, right? See, I'm a scientist, but I actually present the work of my students or my lab members. And in fact, they have far more knowledge on technical and practical aspect than me because I don't work on the bench. So they would be here, and I'm sure they would be happy to take any kind of questions. And I encourage, please don't hesitate to ask even the basic, very basic questions, whatever questions you have in your mind, because time frame I could see it's 2 to 4.
Yeah.
And I asked my postdoc to at least give me 1 hour. So he would be available for 1 hour to answer any kind of questions, starting from the primer designing to whatever questions you have in your mind. Please feel free to ask them. Okay.
Thank you, Dr. Giri. I really appreciate your efforts in making sure that your lecture is presented on time.
Yeah,
Really, really a big
yeah,
applause for this.
yeah. Thanks to technology, now we can record our talk as well.
Yeah,
All
thank
right,
you.
so I'll start. Give me a minute. Let me set up
Sure.
my slides. So I'm using this Google Meet for the first time. So how do
Okay.
I? It's
So on
present.
your screen, sir, you'll see three dots on the bottom, more options.
Yeah, yeah, yeah.
And
And
before
he.
that, there is a present now up arrow.
All right. So let
So
me.
if yeah, so you'll have to open your video recording on your screen, and when you'll present, then you will be able to see that video recording.
All right. So can you see my screen?
Yeah.
All right. So I'm muting myself so that I can play my video. Did you listen to my slides?
Yeah, there is a background music going on, sir.
Because somebody has switched on their mic. Monisha
So
has to
I
mute.
request every one of you, please mute yourself. I mute. I'm muted now. Yeah.
Yeah, yeah. But did you listen to my slide? I just played the first slide.
No. So if you can play again.
All right.
No, we can't hear, sir.
You don't listen?
No, we can't hear. We can see the slides, but we can't hear anything.
Oh, maybe uh, to do this. Shall I email you my presentation and maybe you can play from your computer?
Yeah, I'll try.
All right. So only thing that the moment you click on the slideshow in the PPT,
Okay.
it will start
Mhm.
automatically playing the recordings.
Okay.
I'm quickly sending it to your email address.
Okay.
Okay, I sent it to you.
Okay. I should be receiving anytime. I think it's a big file, so taking a little time to come to
Yeah,
mind.
it will take few seconds.
Yeah, yeah, got it. Yeah. So I'm downloading the file so that there is no interruption during the play. Uh, so I can see the slides, but I again I cannot hear anything.
Did you click on the slideshow option?
Yeah. So I'll share my screen here. Yeah. So here,
Yeah, go to the first slide.
yeah.
Yeah, now hit this slideshow option because I could see the speaker uh, sign
Yeah,
there.
there is a speaker icon here. So I will just see if there is any other option. Yeah. So should I click on the speaker icon? No.
No, this will take you to the next slide. Oops.
I think is there any other way that we're missing? Anyone from the audience can help us?
Ma'am, can I suggest something?
Yeah, yeah, please.
Good afternoon, ma'am.
Good afternoon.
Actually, do one thing, share, open the file in a separate tab
Okay.
and then share the tab. The audio will be coming then.
Uh, so like,
Like in a Google Chrome or something, open it in a separate tab and then share the tab.
okay, like this.
Yeah. Then go to the Google Meet.
Okay.
There in the share option where you get the lower three dots.
Mm-hmm. No, it's not
So there,
about the problem of presentation. It is about there is a audio which is linked to this presentation.
ma'am, do that, ma'am. The audio will come automatically.
So this way.
Yes, ma'am. While sharing, instead of sharing your window or sharing the screen, share tab in
Mm-hmm.
the Google Meet. Where the sharing
Share
option
tab.
comes in the
Okay,
Google Meet,
okay, got it, got it,
share
got it.
tab.
Actually, you're saying share tab.
Yes.
Okay, got it. This is
Is
fine.
it coming now, ma'am?
Yeah. Mm. think फिर से presentation करें। ये presentation है, ये download हो जाएगा। I think up tab share करते हैं। खोल दिया। विंडो। बट इसके साथ ना साउंड है, रिकॉर्डिंग है गिरी, नहीं किया हुआ है बट वो आ नहीं रहा ना।
Dr. Meghna, without sharing it, you just play it once from the downloaded file and just see if you can hear the voice.
Okay, I'll try that also. No, Dr. Giri, I can't hear anything.
Are you using Mac laptop?
No, it's not a Mac one.
It seems the problem is because I did this in Mac.
Oh, yeah, could be that.
Uh,
So,
Let me just try one more way of doing
yeah.
it.
So if it is working without sharing on your computer, sir?
Yeah, I mean, just if you can hear me.
Yeah.
Do you hear this?
No, sir.
You are not hearing anything?
No. Audio.
Because here I can hear my presentation very well.
Okay. So,
Let me just.
what other option we have? Like, so, Dr. Giri, if your postdoc students, they can help to present this presentation?
Let me just ask if they can fix this problem.
Yeah.
Yeah, I just called a computer expert.
Okay, sir. All right. Yeah. We all are still evolving and learning a lot about digital usage and digital platforms.
I switch on this. What is this? Share event is ignore.
No, sir. Just sir, no, for the presentation.
Do you hear me now?
No, no, sir.
Vandit, this is my recorded presentation. Now I am playing this in the Google Meet. But they can see my slides, but they don't see my voice. I mean, they don't hear my voice.
We can hear your voice, sir.
At now,
You can,
now when I'm speaking, actually, it is not coming from the slides. So what to do?
And multiple windows are open. I think multiple applications you have opened, sir. Just
so Dr. Giri, is it possible like any other time when when we can reschedule this meeting?
Just let me try for 5 minutes because
Yeah.
if not, then if we just play here,
Yeah.
we just play here. So it's perfectly fine. But when I'm sharing it, why they don't hear my voice? Here is Google Meet.
go to Google Meet, then share. Then share.
It's a laptop. There's some issue.
There's some issue with Google Meet.
So I think in the meantime, Dr. Giri is taking care to share the presentation. So what I'll do is I'll share a link for your attendance. In the meantime, you can fill your link for the attendance and let us know that you joined the first and the second session.
Yeah, Dr. Meghna, I'm really sorry for this. Just give
No,
me
no problem, sir. No, it's
a
okay.
couple of minutes. I think we would be
Yeah,
able to sort it out.
I understand. It's okay, sir.
Dr. Madhu, please start the session.
How many participants are there for this session?
Yes, sir. Now we can hear.
That is why another meeting started.
Oh my, okay.
Uh,
And the type of radioactive sources that you are using. Hello?
So please, all the participants, mark your presence in the Google form that I've shared. So the link for the form is in your chat window. Please access that link to mark your presence for this session. And I can see 44 participants have submitted, 45 participants have submitted their response. Request others to please submit.
Dr. Meghna, we are trying with a different computer. Please let me know if it works.
Okay.
Share.
A window. This thing.
We'll discuss basics of gene editing using CRISPR-Cas9.
Yeah, we can hear, sir.
Some of our fantastic using
Yeah.
the system. So I'm
Good
stopping it.
efforts.
Yeah,
Very good.
and then I'll start in a minute, right?
Yeah, yeah.
Oh, finally. Thanks to Pavandeep.
Thank you.
You can do it here itself. इसको बंद कर।
This clearly is missing.
Somebody has switched on their
Okay.
microphone, I think.
I request all the participants, please put yourself on mute so that the presentation is audible and everyone can hear without disturbance. Please put yourself on mute.
Okay. So, all right.
Yeah.
So I'm starting it now.
Thank you.
Just I give a introduction. Maybe about
I will discuss basics of gene editing using CRISPR-Cas9, and then I will show you some of our results using this system on its application in improving phosphate use efficiency and transport in plants, especially in rice. So what I'm going to talk about today is I'll start with the basic understanding on how CRISPR-Cas9 works, how this system is modified to further increase its application. There are hundreds of examples of successful use of gene editing using CRISPR-Cas9, but I'll discuss a few, especially in the context of multiplexing, where we can target many genes simultaneously. Then I'll touch upon on screening and obtaining marker-free gene-edited plants. I'll discuss how we can do this. Before I'll move on to some of the results from my own research on use of this technology in transcriptional and post-translation regulation of gene functions. I'll make sure that we have sufficient time for question and answers. So development a new crop variety is always a need, but it takes a whole lot of time, more than 8 to 10 years. In 80s, transgenics were proposed, alternative to classical breeding, and it was expected that transgenics would really enhance the development of new varieties. But transgenics are not accepted worldwide, including India, due to various reasons. And now CRISPR-Cas9 has emerged, or it is proposed as a new alternative to transgenics and classical breeding. And this system is more precise and it's very rapid, fast. It just takes half of the time of what is taken in the production of a new variety if we know the gene of interest we are targeting. And we also expect that gene editing will allay some of the fears associated with the GM crops. So let's start with the basics. I'm sure most of the people attending this talk They already know the basics of CRISPR-Cas9.
So, Mr. Pavandeep Singh, can we put it on the slide mode?
So what is CRISPR? CRISPR is clustered regularly interspaced short palindromic repeats present in bacteria, E. coli. They were discovered in E. coli. What clustered means basically that they are present in the genome in the clustered form, and these are about 40 nucleotide long sequences. If you read from each side, they read same. So when their transcript is produced, their transcript form a hairpin, and they are all identical. So you have the repeats, 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 and repeats. But they're also interspaced by sequence which we call as a spacer DNA. And what is unique about this spacer DNA is that these spacer DNAs are not identical. They are all unique. So in the bacterial genome, we have repeat, spacer, repeat, spacer, repeat, and spacer, and so on. In addition to this, the close to CRISPR locus, there is another locus which encodes the Cas protein. So Cas stands for
So
CRISPR.
if anybody is hearing me from Dr. Giri's side, please, please see if the slides are moving.
This Cas endonuclease is a type 2 endonuclease which has both helicase and DNA nuclease activity. So it can unwind the DNA or it can cut the DNA.
Excuse me, sir.
That
Sorry, sir.
an integrated part of.
Hello.
Yeah.
Yes, yes.
Okay. Sorry, we were not able to see the other slides, so it was stuck on the first slide. That is what we wanted to convey.
Oh, you can fix this. I will I'm sure most of the people attending this talk start from the first slide. Yeah.
Also, if you can put it on slide mode, slideshow mode, so full screen basically.
So what I'm going to talk about today is, I'll start with the basic Just a second.
Yeah, no.
Is it working now?
Yeah.
Just
Yes.
So what I'm going to talk about today is, I'll start with the basic understanding on how CRISPR-Cas9 works. It's working fine? How this system is modified further increase its application. Let me see. There are hundreds of examples of successful use of gene editing using CRISPR-Cas9. But I'll discuss a few, especially in the context of multiplexing, where we can target many genes simultaneously. Then I'll touch upon screening and obtaining marker-free gene-edited plants. I'll discuss how we can do this. Before I'll move on to some of the results from my own research on use of this technology in transcriptional and post-translation regulation of gene functions. I'll make sure that we have sufficient time for question and answers. So development a new crop variety is always a need, but it takes a whole lot of time, more than 8 to 10 years. Is it working fine now? In the 80s, transgenics were proposed alternative to classical breeding.
I was saying if it is possible to put it on full screen mode,
enhance the development
the
of
slides on full screen mode.
But transgenics are not accepted worldwide, including
But we can see the Microsoft
and now
PowerPoint window here,
first, or it is
rather
proposed.
than the whole full slide mode.
So development in new crop variety always
Wait now.
but it takes more lot of time, more than 8 to 10
Is it full
years.
screen now?
Yeah, it is full screen and it's clearly audible.
Native to
Fine.
Thank you.
classical breeding. And it was expected that transgenics would really enhance the development of new varieties. But transgenics are not accepted worldwide, including India, due to various reasons. And now CRISPR-Cas9 has emerged, or it is proposed as a new alternative to transgenics and classical breeding. And this system is more precise and it's very rapid, fast. It just takes half of the time of what is taken in the production of a new variety if we know the gene of interest we are targeting. And we also expect that gene editing will allay some of the fears associated with the GM crops. So let's start with the basics. I'm sure most of the people attending this talk, they already know the basics of CRISPR-Cas9. Let me introduce it again briefly. So what is CRISPR? CRISPR is clustered regularly interspaced short palindromic repeats present in bacteria, E. coli. They were discovered in E. coli. What clustered means basically that they are present in the genome in the clustered form, and these are about 40 nucleotide long sequences. If you read from each side, they read same. So when their transcript is produced, their transcript form a hairpin, so which is called as a palindrome as well. Now these sequences are present repeatedly in the bacterial genome, and they are all identical. So you have the repeats, and repeats. But they're also interspaced by sequence which we call as a spacer DNA. And what is unique about this spacer DNA is that these spacer DNAs are not identical. They are all unique. So in the bacterial genome, we have repeat, spacer, repeat, and spacer, and so on. In addition to this, the close to CRISPR locus, there is another locus which encode the Cas protein. So Cas stands for CRISPR associated genes. So these Cas9 encode an endonuclease, and this Cas endonuclease is a type 2 endonuclease which has both helicase and DNA nuclease activity. So it can unwind the DNA or it can cut the DNA. So it turned out that this system is actually an adaptive immunity for bacteria against viruses. So if when a virus is attacking a bacteria and bacteria doesn't have a system in place to check the viral infection, the viral will keep on replicating and ultimately it will kill the bacterial cells. But with this system in place, this Cas9 protein is produced, and from here the CRISPR RNA is produced. This CRISPR RNA has a sequence which has the homology with the viral genome. So part of the viral genome. So this complex will target the viral DNA and it will destroy it completely. What if the virus is new and it doesn't have any homology in the bacterial genome? So in this case, another class of Cas protein is produced, which is class 1 Cas proteins. These Cas protein chew the viral genome into small pieces and integrated part of the viral genome in the bacterial DNA. So it becomes a memory for the future infection with this virus. So if you see that this system is nothing but this is an adaptive immune system in bacteria against the viruses. And all these spacer sequences, these are essentially history of past infection of the viruses. Now, with this history of this past infection, now bacteria is able to defend itself against the viruses. Now, in addition to Cas9 and CRISPR locus, bacterial genome also produces a RNA, which is tracer RNA or trans-activating RNA. Now, this RNA is not the part of CRISPR-Cas9 locus. Rather, it is produced quite away from this locus. And this tracer RNA, it forms a hairpin with the 3' end of the CRISPR RNA. So the 5' end of tracer RNA and 3' end of CRISPR RNA, they join and form a hairpin loop. Now, this hairpin loop formation is essential for the activation of Cas9. So binding of these components, so it's basically a 3-component system where we have a Cas9 endonuclease, we have CRISPR RNA, and we have a tracer RNA. When CRISPR RNA and tracer RNA, they form a complex and they bind with the Cas9. Cas9 becomes active for degradation of the incoming viral DNA. Now, in addition to this, the tracer RNA and Cas9, they also help in CRISPR RNA processing. The initial CRISPR's RNA size is quite big, and the mature CRISPR RNA is only about 42 nucleotide in the native bacteria, in the natural conditions. So this protein Cas9 and tracer RNA, they also help in the CRISPR RNA processing. Now the question is that why this system is so much used in nowadays. So these two ladies, Jennifer and Emanuele, they are working on this bacteria, Streptococcus pyogenes, and they were working on Cas gene. In this bacteria, Cas9 is endonucleate, which is produced by a single gene. So they have demonstrated many things of this system, but the key aspect of their discovery and applications are, the first thing they showed that tracer RNA and CRISPR RNA, they can be combined in a single RNA molecule, and that molecule is known as guide RNA. In bacteria, tracer RNA and CRISPR RNA were coming from 2 different locus. But they have shown in vivo and in vitro that CRISPR RNA and tracer RNA, they can be combined, and this molecule can work perfectly fine as it works in the native system. Second thing they have done, that they have shown that this spacer RNA, which was basically having the homology with the incoming virus. In place of this sequence, you can put any sequence which has the complementary sequence in the any target genome. And this system then can target any genome, any gene for double strand break using this Cas9. So what they have done, they have converted that 3-component system into 2-component. So one is guide RNA and another, in place of spacer sequence, we can have the sequence of our gene of interest. So if you see that all the vectors we use in the gene editing, this part is already there in vector. It is already synthesized and it is incorporated in vector. Only thing that we have to add is this guide RNA or this sequence, this hardly 20 nucleotide long sequence, which is complementary to any gene in the genome. So they have simplified this system. Let's understand a little bit more detail about the system. Here we have marked all the domains of Cas9. Before that, let me introduce the concept of PAM. So what is PAM sequence? So PAM sequence is proto-spacer adjacent motif, and for Cas9, PAM is NGG. So basically, and can be any nucleotide. So PAM is basically where Cas9 binds, where Cas9 starts unwinding the DNA and cutting the DNA. So generally, 3 to 6 base pair upstream to PAM is the site where Cas9 cut the DNA, both the strand. That means it makes a double strand break. So here are the different domains of Cas9. One more thing, that activation of Cas9 requires the binding of both guide RNA with the Cas9 protein. So this activation brings the conformational changes in the Cas9, and then it becomes active. So this GG nucleotide in the PAM sequence, they are targeted by arginine of the beta helix of Cas9, where this strand separation starts, and this causes the local strand separation and binding of the guide RNA starts. The base pairing between guide RNA and target strand progress and forms a guide RNA and target heteroduplex. Now here again we have defined the different domains of Cas9. This PI, the C-terminal motif of the Cas9, is the site which recognizes the PAM sequence. And once the guide RNA form a heteroduplex with the target DNA sequence, the 2 nucleus domain of Cas9, they cut the respective strand. The HNH domain, it cuts basically the strand which is complementary to guide RNA, while RUVC nuclease domain cut the PAP carrying domain. So this double strand break is repaired by two mechanisms. One is NHEJ, which is non-homologous and joining method in the cell, where cell join the two end by randomly inserting or deleting few nucleotides. So this results in the random insertion or deletion and thus of nucleotides and thus induction of a mutation. Second method, which is less pronounced, is HDR. Here cell use the sister chromosome to replace the double strand, to repair the double strand break, and this HDR can be used where in place of the sister chromosome, the cell can use the template that we want to introduce in the gene, in the target genome. So this HDR repair is basically used for gene replacement. So in a mutation, if you see the mutation in the genome is induced by editing, deleting. Editing means the base is replaced. Deletion of bases, addition of bases, or complete replacement of the gene or a fragment of the gene. The basic function of the Cas9 is that it makes the double strand breaks, and which is repaired by an HEJ leading to the mutation. But if we make the either of the nuclease domain non-functional, Cas9 will become nickase. That means it will nick the one strand. It will not cut both the strand, but one strand, and it can be used for replacement of a target gene with the gene of our interest. We can also mutate both the kinase domain, both the nuclease domain of the Cas9, and both of them will become non-functional. And in this case, Cas9 will become a dead Cas9, but it will still retain the DNA binding capacity. It will still bind at the specific position in the genome, helping by the guide RNA. Now, the dead Cas9 has become a perfect delivery vehicle for recruiting the effectors like the transcriptional activators, transcriptional repressors, and other means, so that you can now influence the target gene expression and its activity. So let me summarize this whole system quickly. So we have a single guide RNA, which carries the gene, the sequence which is complementary to the gene of my interest. It carries the Cas9 protein. The Cas9 protein and guide RNA, they form a complex. The complex goes through DNA until they find a match close to the PAM sequence. That leads to the conformational changes that activates the nucleus. Double strand break is created, and that double strand break is repaired by non-homologous end joining, resulting in the mutation. Now, if we have clone our guide RNA in a plasmid, it can be easily introduced using agrobacterium-mediated system into the plant cell, or we can use the plasmid-free guide RNA using simply DNA or RNA, and we can have the bacterial purified protein. We can make a complex of them, can coat gold particle and introduce them into a cell, or we can use viral vectors. Now, once inside the cytosol, this system can be used for organelle gene editing, like the plastid and mitochondrial gene editing. It can be used for RNA editing. And then once inside the nucleus, Cas9 can be used, this system can be used for gene knockout. It can be used for precise gene editing, like the base editing. If we have fused the base editor with the Cas9, it can be used for HDR, gene replacement, or it can be used for transcriptional activation or regulation if we are using dead Cas9. Now, most of the Cas9 and its variants and other endonucleases of this class, they are active about 30 degrees Celsius. And this is one of the reasons that gene editing has not been very successful in Arabidopsis, because Arabidopsis grows about at 27 to 28 degrees Celsius, and these proteins are more functional at 30 degrees Celsius. Now, tremendous amount of advancements have been made in this system to increase the specificity of mutation and to increase its applicability. New Cas9 proteins from multiple bacteria have been identified. Cas9 has been mutated in different ways, and new Cas9 with different PAM specificity have been identified. So we no longer need NGG. Now we can target a variety of PAMs listed here. Cas9 has been repurposed with enhanced specificity towards targets by making it more specific to the individual we are interested in targeting for gene editing. And there are many more modifications for its ever increasing applications. There are hundreds of examples where gene editing has been successfully used for improvement of traits in crops. But if I have to choose one, this is the one which I would like to choose, and that is the domestication of wild tomato, which was done by gene editing. So here is wild tomato, and here is a domesticated tomato we eat today. This is a wild tomato, which is very ancient. This tomato has intermediate inflorescence, so no mechanical harvesting is possible. It has a small fruit size, which is not economical. It is also day length sensitive. That means it can grow only in certain area where, depending on the sunlight or day length. But like many plants, the wild plants, they are highly rich in nutrition, and they are also stress tolerant. This domesticated tomato, it got determinate inflorescence. Now the mechanical harvesting is possible. It has large fruit size. It is day length insensitive, so we can grow in larger area. But it got poor nutritional values, and it is extremely sensitive to various biotic and abiotic stresses. Now this is not true for only true for tomato, but this is true for many crops because many useful traits were lost in modern varieties during Green Revolution. But this journey from wild tomato to domesticated tomato, it took thousands of years of natural selection, random mutations followed by natural selection, and decades of breeding to produce a domesticated tomato with the beneficial traits. But this journey of thousands of years was reduced to just 3-4 years by this group, the Lee et al., where they have targeted 5 genes which are encoding different traits which are useful to produce a domesticated crop. And all these 5 genes they targeted in a single exercise of making for gene editing plant. They converted this wild tomato into the cultivated tomato. And while doing so, they targeted coding sequence, they targeted regulatory regions, cis regulatory regions, they also targeted upstream ORF of these genes. So while they have introduced new traits into the wild varieties of tomato, they could retain the traits like the tolerance to biotic and abiotic stress. Has also achieved the same. These two people came and they could also get a tomato in the single event, hardly in the 3 to 4 years. So you can full application of this technology, where that journey of thousands of years can be cut short to just 3 to 4 years. But both of these groups, they smartly targeted monogenic traits. That means the most of the traits they have targeted, they were encoded by a single gene. So when a trait is encoded by a single gene, it's become easy to change that trait or edit that trait. In case of polygenic traits, where a trait is influenced by multiple genes, it becomes really difficult to edit them in one go. But to me, these are the kind of a path-breaking discoveries of use of gene editing in crop improvement. And this has opened something which I call as a neo-domestication, and it has been proposed that we can use this technology to domesticate many other wild relatives of the other crop plants. Because most of the wild relatives, although they have some undesired traits, but most of them, they have these traits of tolerance to biotic and abiotic stresses. And breeders always, they look for wild relatives to good genes which can be introduced into the modern varieties. But it's a very long process. For example, in rice here, we can introduce these traits. This is wild rice, this is the cultivated rice, like the erect necks, increase trailer number, optimum flowering time and ideal plant architecture, and while we can still retain the tolerance to biotic and abiotic stresses. And this has been in fact achieved recently. Wild rice has been converted into a domesticated rice in the single event using gene editing. So these are the wonderful applications of gene editing in the plant improvement or crop improvement. And all these applications were possible only because of one unique, I would say, the application of gene editing, which is multiplexing, where we can target multiple genes simultaneously. So if we can have one guide RNA targeting one gene, we can have a second guide RNA targeting second gene, and so on. Now, one way of doing it, or initially how people did it, actually they used individual promoters to express a guide RNA. Or we can, alternatively, we can use completely DNA-free multiplexing, where simply we have guide RNA, we have Cas9 protein, and we can directly bombard gold particles carrying this complex into the cell. Now, alternative to this, and which has been quite successful, is this technology, where guide RNA are cloned as a polycistronic transfer RNA guide RNA unit. Here, multiple guide RNA can be cloned under a single promoter, and these guide RNA are interspaced by transfer RNA. So this is produced as a single transcript, which is then processed, and individual guide RNA can be released, which can be bind with the Cas9 protein to target the different genes. I'll show you the application of both of these methods. So here in this example, they have used 5 different promoters to target 6 guide RNA, sorry, and they have used individual promoters. So in this case, the first difficulty is that the cloning becomes cumbersome, because for transcription of multiple genes, it will involve the multiple steps. Second issue is that the vector size becomes very big, so it is not easy to transform into the plant cell. And also, when they did the gene editing using such a construct, such a large construct carrying different promoters, the maximum editing rate with multiplex editing of a single target was 56%. So where they have targeted a single gene, it was 56%. But where they have targeted multiple genes, it has come down to around 10%. So this is not very effective if we are doing multiplexing. Now, alternative to this, as I just explained, is this system, which is polycistronic transfer RNA guide RNA. Here, one guide RNA is cloned, then we have a transfer RNA, then we have guide RNA 2, guide RNA 3, transfer RNA, like this. Now, they all can be expressed under a single promoter, and this would be the primary transcript having both guide RNA sequence and transfer RNA sequence. And now the cellular machinery will process this transcript, and they will remove the guide RNA from this transcript. So now each guide RNA is free to combine with Cas9 protein and to target the target gene. So here you can have n number of guide RNA targeting n number of different genes. And this system is very easy to assemble. Just in 1 and 2 steps using golden gate assembly, you can assemble multiple guide RNA in a single vector, and size is not that big. And results are very good. For example, PTG7, this construct is using 7 guide RNA. And if you see in some cases, the editing efficiency is about 100%. So PTG-based CRISPR construct, they showed highest efficiency in multiplex gene editing, where non-PTG based has low multiplexing gene editing efficiency. So the conclusion here is that we can express multiple guide RNA under a single promoter, and they work fine. Now, while the single base pair editing or multiple single base pair, single gene editing or multi-gene editing has been achieved in rice, what has been always a challenge in plant is the gene replacement, plant or even in the higher eukaryotes, because they have low rate of HDR compared to NHEJ. For gene replacement, we need HDR. And this HDR is not only in low rate, but is only active during the short span of life cycle. Second, HDR does not change the target sites because this is a perfect replacement. So it may be targeted, those sites may be targeted by guide RNA Cas9 complex again. NHEJ may also work for gene replacement, but accuracy would be very low because NHEJ is a error-prone method. So at the junction, we will always have the error. Now this group has smartly used NHEJ for gene replacement. So what they did that instead of targeting exons, they targeted introns. Now even if there is an imperfect repair, the intron would be spliced out, so the gene function would not be affected. So what they did that they used this mutated exon 2 and they cloned the guide RNA target sequence flanking this exon 2 and cloned them in a vector, and they co-bombarded this Cas9 carrying vector and the vector carrying the exact target site of the target gene and also the insert. And they bombarded it into the rice, and they found that 8 genes were perfectly replaced in 390 T0 transgenic lines. Now this frequency is small, but it is good enough. 2% is good enough in case of rice. And all the plants carrying the mutated exon, they showed herbicide tolerance. So this is the example that even we can use NHEJ method for gene replacement, but we have to be careful that instead of targeting exon, we can target the intron. Now let me shift to some of the work we are doing in this subject in my laboratory. I work on improving phosphate use efficiency in rice. Phosphate is very important for plants because plants cannot grow without phosphate. And in past 7-8 years, we have identified some very important genes which can be targeted for improving phosphate use efficiency in rice. And mainly we have used a transgenic system for showing the efficacy of those genes which we have identified. For past 4 years, we have started using gene editing. And today I'm going to show you some of the examples which we have, where we could have demonstrated the use of gene editing for improvement of phosphate use efficiency in rice. So one of the theme in my lab is the membrane repeatedly modeling. Let me just explain this very briefly. So membranes have phospholipids, and phospholipids mean phosphate. So what happened when plants are grown under phosphate deficiency? These phospholipids are hydrolyzed and they are replaced with galactolipids. Now these galactolipids, they do not have phosphate. They are simply galactose containing lipids, but they can perfectly replace the part of phospholipids. So in a way, plants have used some of the phosphate which is released from the hydrolysis of phosphorus for carrying out more important functions. And this loss of phosphate or loss of phospholipid can be compensated by galactolipids. Previously, we have shown we have characterized some of the genes of this pathway. For example, here is one gene. This is GDPD2. This gene encodes a glycerophosphodiester phosphodiesterase, which helps in the solubilization of phospholipids. So when we overexpress this gene, the transgenics, they showed longer roots, while the RNA lines, they showed reduced roots. But these transgenics, they showed more galactolipid accumulation, and they also showed more phosphate accumulation under phosphate deficiency condition. So such genes of which are involved in the membrane repeatedly modeling, they can be targeted for improving phosphate use efficiency in rice. So then we started using gene editing, and we have identified a gene which is MGD3. This gene is responsible for the galactolipid biosynthesis, that phosphate-free lipids I just discussed with you. So we targeted its first exon for the mutation using guide RNA targeting to its first exon. If we target the beginning of the gene, then the chances of getting mutations are always high. So we got mutations, and if you can see, there are different types of mutations which we got in this gene, and our mutation frequency rate was very high, which was 88%, and we used the same PTG system where transfer RNA are cloned along with the guide RNA. And in some cases we got very big deletion, but in some cases we got just one base pair deletion in the gene, and that was leading to the stop codon formation. So the gene was completely knocked out. So we selected those lines where we had only single base pair addition or deletion, because they are because if the deletion is big, those things are not preferred. So in the second generation, we found biallelic homozygous mutation. So let me define this. If the mutation is only in the one allele, it is monoallelic. Another allele of the, because rice is a diploid, so another allele could be a wild type. If both the alleles are mutated, it is biallelic. And both the alleles can be mutated in different base pair. So then it becomes the biallelic heterozygous. So key here is to identify biallelic homozygous. That means both the alleles, they are carrying the same base pair deletion or addition, as you see here. Either A is edited or T is edited. So we selected these lines which were, for example, here 38 and 52, which were biallelic homozygous for the mutation. Now our next target was to find a marker-free line, because Cas9 and guide RNA, they will keep on mutating genes which are showing some homology, so we will have many off-target mutations. And this system, CRISPR-Cas9, is a very good system for raising marker-free gene editing lines, because the tDNA carrying Cas9 will integrate at a certain part of the genome, but they will act at some different locus in the genome. So the site of integration of tDNA and the site of mutation would be different. These two locus will segregate independently in the subsequent generation, and this gives an opportunity to select tDNA free but mutated plant. We can also get marker free plants using the DNA free gene editing, where we do not use DNA at all, rather we use artificially synthesized or in vitro transcribed guide RNA, and we can use the bacterial purified Cas9 protein. So then we can coat this guide RNA and Cas9 on the gold particle, and we can bombard in the rice calli, and the plant will be formed. So benefit here is that guide RNA protein, they will not be integrated into the genome. They will be working transiently only. So since there is no transgene integration, so there would be no carryover of transgene in the next generation. So both guide RNA and Cas9, they would be destroyed in the first generation itself after just some time. So, but the problem of this system is that this system got a very poor mutation frequency. So we chose to use the tDNA segregation based method. Here the key is that if you have a single copy insertion line, which you can identify using the southern blot or hygromycin resistance segregation, any antibiotic resistance segregation which is there in the tDNA. And then this single copy line, this will segregate genotypically into 1:2:1. One would be the plant which will not carry the tDNA at all, but it may be edited or it may not be edited. So we have to identify this plant. So to do so, we grew these plants without any antibiotic selection. And as you see here, this plant is not carrying tDNA, but this is mutated. And when you grow seeds of such plant on the hygromycin, any antibiotic selection, they do not grow. That means these plants are not carrying tDNA, but they are mutated. And we have seen that this mutation is heritable at least for 3 generations, and this is just one base pair insertion. So it is as good as any EMS generated mutant. So we used these knockout lines and found that since we have knocked out this gene, so galactolipid accumulation was poor under -P, which resulted in the poor phosphate use efficiency. So this is a demonstration that just single base pair change in this gene rendered it's non-functional, and it results in the reduction of PUE, phosphate use efficiency. This is just showing that we have also overexpressed this gene and proved that our phosphate use efficiency is increased when we overexpress this gene. Now, all the data I have shown you, that was achieved using the DNA sequencing, but many labs may not have access to DNA sequencing. So for the detection of mutant plant, we can simply use PCR using this another technology which is called PCRE. Here the key is that you select a PAM in the region where you have restriction site for an enzyme. So for example here ACU1, if this site is mutated, so the mutated site will not be digested by this enzyme. We identify simply using restriction based digestion. For example here, this is first is wild type, which is completely digested, and here are the mutant lines which are resistant to digestion. So using this method also we identified marker free lines which were gene edited. Now let me change the topic for a little bit. We all feel stress, panic and anxiety, and along with the stress, panic and anxiety, they come uninvited, and they hamper our ability to cope up with the stress. And what all the gurus, they tell that you have to separate stress with the panic and anxiety so that you can find the solution. Now, it is common that we all feel stress, but do plants feel stress? And answer is yes. Not only they stress, rather they panic under phosphate deficiency. Here is the rice plant growing in sufficient phosphate. Here's the rice plant growing in deficient phosphate. And what was found that these plants are not so poor because of phosphate deficiency only. Rather, there are thousands of genes which are induced under phosphate deficiency, and they got nothing to do with phosphate deficiency response. So that is an unnecessary burden which is resulting in this growth dwarfism in rice. Also, rice, the plants under phosphate deficiency, they are so desperate that they suppress their immunity response by suppressing this transcription factor PHR2. So why they suppress their immunity response under phosphate deficiency? Because plants want to attract more and more microbes, because microbes help in the phosphate solubilization. So in order to get more phosphorus, plants are giving up on their immunity. What is also found that in plant cell, phosphate levels are really, really high. They are in the micromolar range, but in the soil, it is hardly in the micromolar range. In the cell, it is in the millimolar range. But plants do not need this much phosphate. So if it somehow we can remove that panic part, we can cut down the use of phosphate by plants. And we have some evidence from Arabidopsis. In Arabidopsis, this transporter, phosphate transporter, it expresses in the root in normal plant, and it transports phosphate from root to shoot, so plant can grow normal. In the mutant of this gene, all the phosphate is locked up into the root, and shoot part is not getting phosphate, so shoot part is dwarfed. What was found that this gene got 3 domains, domain 1, 2, and 3. And if you complement this mutant with the different domain, so when you complement this with D3 domain, the plants still have half of the phosphorus compared to normal plant, but they almost grow normally. So this is interesting because this is showing that phosphate level alone are not responsible for growth retardation under phosphate deficiency. But this D3 domain, it lacks the phosphate transport activity, and that is why the phosphate levels are high in the complemented line. Now, one way could be that you overexpress D3 because D3 is somehow helping grow plants normal despite having the low phosphorus. Or you can express the D3 under the native promoter if you can delete these 2 domains, D1 and D2. Now the question is how gene editing can help here. So generally we use this multiplexing system for targeting the different genes by targeting different guide RNA to some part of the gene. We can also target 2 guide RNA to 1 gene. So for example here, this guide RNA will bring Cas9 here, this guide RNA will bring Cas9 here. They will make a double strand break. So part of the gene 2 would be deleted and remaining 2 part will join, and we have a small gene. So using this technology, this application of gene editing, we thought of deleting domain 1 and domain 2 by targeting introns. Again, we targeted introns. Exon targeting means actually is a problem. So if my guide RNA is working perfectly fine, this will take Cas9 here, this will make a cut, and I will have a truncated but functional protein because I kept the ATG of the domain 1. Benefit here is all the transcriptional regulatory controls are intact because I have not done anything with the promoter and there is no need of making overexpression transgenic. But the challenge was to make a 3.8 kb fragment deletion. And deletion of such a fragment is not reported well. We screened more than 150 lines, but we could get a line where this fragment was completely deleted. And when we tested these plants, we found that phosphate levels are low and plants, they grow normal. So we have basically achieved what we were intended to do, that if we can reduce that panic part in the plant, it can reduce the dependence on the phosphorus, although this is very preliminary data. Next, I can show you an example how we have used this CRISPR-Cas9 to alter the transcriptional regulation of phosphate transporter, the same transporter. This transporter has the W box, which are the Werke transcription factor binding site in its promoter. But these Werke transcription factors, they are repressor of this gene under normal condition, but in phosphate deficiency, these transcription factors are degraded, so it cannot repress the function of the it cannot repress the expression of forward. So we thought of what if we remove this W box binding site and then this transcription factor, this repressor will not bind to the W box and this gene will show the high expression. We can also achieve this by knocking out those transcriptional repressor, but that will result in the undesirable effect. We can also overexpress this transcriptional this transporter, but that will also result in the undesirable effect. But if you simply modify these acting elements, then we are affecting only this gene and we are not affecting other gene. So to achieve this, we designed 4 guide RNA targeting these 2 W boxes, and we wanted to delete all of them. We could achieve the deletion of one box, we could not achieve the deletion of both the W boxes, but deletion of one box is achieved. And we have seen that the expression of this gene has increased up to a certain level, and so is the phosphate level also. So what I have shown you here is that we can control the undesired effect of a gene repression or gene overexpression if we directly target the promoter of a gene and we don't choose the overexpression or underexpression of a gene using RNAi or overexpression methods. I'll stop here, and in summary, I want to tell you that CRISPR-Cas9 system is a versatile system which can be used in multiple ways. In most of the labs, it has been used only for gene knockouts. So it shall not be restricted to gene knockouts. It can be used for multiple applications. Few of them I have shown you, and plenty of them are available in the literature. One thing which is lacking in the most of the reports is the data of targets. Cas9 and guide RNA always have some similarity in the genome, and it can bind with the other genes. So we need the data of targets of target sequencing in these gene-edited plants just to make sure that the editing is specifically only to our gene of interest. What we have also seen, and we have shown that to minimize the off-target mutations, one is we don't use the DNA at the first place. We can simply use transgene-free gene editing, and we can also use this approach of marker-free gene-edited lines by simply segregating out tDNA, because this is one of the options to minimize the off-target mutations. Lastly, I would say nowadays, so many people are talking about CRISPR-Cas9, and at times I found that people are actually exaggerating the applications of Cas9. It got unlimited applications, but CRISPR-Cas9 is not a panacea to agriculture problems. The classical breeding and transgenics, they will still remain there. CRISPR-Cas9 is one additional tool which is a little bit more specific and more faster, so it should be made use in that way only, because if you project too much, then it creates problem. Before I acknowledge my lab, I must show you this picture. Here in this picture, a strong radiation agent is kept in the center, and you have these different crops in the concentric rings. This radiation bombard these crops or their genome brutally, and we do not know what kind of genomic aberrations are happening there, what kind of changes are happening there. And this is the food from such crops which end up in our table we are eating, because atomic gardening or breeding is allowed. Unfortunately, gene editing, which is very precise, and I have shown you example that my plants are completely free of tDNA, there is no tDNA, and there is only one single base pair change, but that specific plant is not allowed. Gene editing is being treated as GM only in India, although some discussion is going on where, in fact, in many countries now the gene edited plants have been approved and they have been allowed to, farmers are allowed to grow. In India, there is still discussion going on, and although we could not do anything on GM, but I hope that on the gene edited plants, government may allow their limited use at least. All the work I have shown you, this was not done by me, but it was done by the fantastic group of people I have in my lab. The gene editing work in my lab was initiated by Lokesh. He has just finished his PhD. He is now postdoc in the lab. And this, the panic part removal from the rice plant towards the phosphate deficiency was done by Kanika and Balaji. I must acknowledge generous funding from Ministry of Science and Technology and my parent agency DBT, my own institute, and IPGR. Thank you very much. I would be happy to answer any questions if you have. Thank you.
So, thank you, Dr. Giri. I think it was an amazing, awesome presentation of how CRISPR can be used in different plant varieties. So we learned what CRISPR-Cas is, how CRISPR-Cas system works. We also understood through different examples that how gene editing can be done in plants, and how de novo domestication of wild tomato is done using this gene editing technique. Also, a great presentation that tells us about use of CRISPR-Cas to improve phosphate use efficiency in rice plants, and how phosphate-associated stress in plant can be reduced using CRISPR-Cas technology. So all these aspects of presentation are very different from each other, but all took us to one point, that how CRISPR-Cas can be widely used and it is applicable in different plant varieties, different kind of problems that we are facing with crops can be resolved using CRISPR-Cas technology. So thank you so much, sir. I don't know if you are here listening these words or not, but maybe somebody from your lab is who can convey our regards and our gratitude for presenting this great information to all the audiences.
Dr. Meghna,
Yes, sir.
I just joined. Thank you very much.
Okay.
And now I have with me Dr. Lokesh and Nika.
Okay.
These are the two who are actually working on the bench with gene editing, and they would be really happy to answer any kind of questions your audience have.
Thank you so much, sir. So I've already met Dr. Lokesh. I think Dr. Lokesh, you remember.
Last time we came there. Yeah.
So we have already met, and we already saw his presentation over CRISPR-Cas. So audiences, if you have any question, the participants, faculty members, so please you may ask, or if you're not able to ask through the speakers, so please type your questions here. So there is one question.
I'm handing over this laptop to them. so that I
Yeah.
can rejoin my another meeting.
Thank you so much, sir. We are really grateful. So you have put a lot of effort in recording and then again playing that recording in front of all of us. Thank you so much, sir. Yeah. In the meantime, you please others can type their questions in the chat box. So I'll be happy to read out your questions for the postdoctoral fellows of Dr. Giri.
Hello.
Hello. Yeah.
Uh, uh, I am reading one question that other than phosphate, what are the other molecule influencing editing?
Yeah.
I want to clear that phosphate is not doing the any kind of editing. We are just working on phosphate deficiency. It is the CRISPR-Cas9 system that did the editing. Phosphate
Okay.
have nothing to do any editing.
Yeah. Yeah. So I hope the answer is very clear about this, that how CRISPR is actually doing its work without our interference. So there is one other question, that in between Gibson and Gateway, which could be the more effective system while making the constructs?
For
What was
making
your experience in research on the same?
in my experience, I prefer Gibson assembly because for Cas9 we have very short guideline that we have to clone, and for gateway cloning it is different, and in Gibson cloning you can clone multiple in single go.
Okay. So would you little bit elaborate what are Gibson and Gateway kind of systems? Because I think many of us are not aware about.
Actually, in Gibson assembly, there are type 2s restriction enzymes that cleaves outside the recognition site,
Okay,
and they have another ligase enzyme that ligate the fragment depending on the compatibility of the overhangs.
all right.
But in case of gateway, it is LR based, recombination based. So it
Mhm.
have different requirements, and Gibson assembly can be modified accordingly because restriction site can be removed with the digestion.
Okay, great. So any other question? Yeah, there is one more coming. So phosphate is a major nutrient. Can the same technology be used for studying the effect of micronutrients too?
Yeah, it depends on the gene, whether your gene is working on that micronutrients or not. Our gene that we selected, they are phosphate deficiency responsive genes and already known in different model system that they play role. So micronutrients, if for example they are specific transporter for micronutrients, if we target, we can definitely knock out that gene or edit these genes for micronutrient requirements.
Okay, yeah. So any other question? Please type in your questions, even if it is very basic understanding of CRISPR, because it is a very new technology. Very few people in our country are actually working on CRISPR-Cas. So it is okay to ask any kind of basic concept-based understanding questions related to CRISPR. So one more question here. So from Dr. Nanda, how difficult it is for applying CRISPR into polyploidy plants? What are the major challenges?
In polyploidy plants, actually ploidy level is different, so might be the efficiency of the CRISPR-Cas9 system is not good as we found in the diploid plant where we have to target only 2 copies. So but it is possible because theoretically it can work in polyploidy, but it is difficult in screening also because we have to screen more number of colonies, or depending on the what kind of you are strategy used for sequencing or edit finding out the mutants.
Okay, great. So any more questions? If you want to unmute yourself and ask the question, you're most welcome. So from Dr. Vivek Srivastava, can it ca- uh, can it be used for, or it is an important technique for use in genetic disease of human?
I have no idea. Seriously, I have no idea about that because we are working on plant, and I don't know whether it is applied for human genetic disease or not. I don't have any
Okay.
idea about that.
But I can assure you, Dr. Vivek, in the coming sessions, we will be covering more use and application of CRISPR on different disease conditions, including sickle cell anemia, Huntington's disease, including cystic fibrosis also. So yes, I can say it can be used. But yeah, it is still under research and consideration. So any other question? Okay. So I think everyone has really absorbed and tried to understand different layers of CRISPR-Cas usage in case of plants, as explained by Dr. Giri. And it will take time to understand and then come back with more questions. So maybe I can post the questions to Dr. Giri directly if there are any such later on. So thank you everyone for joining in. Thank you everyone from Dr. Giri's lab to help and support during this time.
Thank you, ma'am.
Thank you a lot. So we look forward to learn a lot again from your lab, or the new information, the new research that is coming out from your lab is useful for us as teachers to impart it to our students that this is what is going on. So thank you so much. So with this, Ms. Rajani, yeah, sure. I'll share the recordings of each and every session within a day or two as I receive the recording from the Google team, because it takes time to receive the recording from Google. So thank you so much. Have a great evening. And with this, we'll close the sessions for today, and we'll meet tomorrow again with as per the schedule that I've already shared with all of you. Thank you so much.